Hello, friends. It's Mr. Rogers here, and I hope that you guys have had a wonderful day. And so we're going to talk about the four evidences of evolution. The four evidences of evolution, we're going to take notes and a piece of paper, and I'd like you to make the piece of paper four boxes like this. You can either do this by just folding it and doing a hamburger fold and a hot dog fold, or you could just draw the lines, or whatever way you want to have four boxes on one piece of paper. And so there's four evidences, obviously, evidence here, evidence here, evidence here, evidence here. Doesn't matter where you put the evidences. You could put the first evidence down here. I don't care, as long as you have all four. So you don't have to write this down. I just want to remind you, evolution is a change in a population of organisms over time. And this change happens in their genes and their genetics. And so we can add the word genetic to that. But truly, like in 10th grade, I teach change in a population of organisms over time, or change over time. Now, there's four types of evidence that we've kind of produced to show evolution of living things. There's plenty more evidences. There's plenty of things and facets that go into evolution. But these four support the kind of ideas that we're going to be talking about in this class. So first one is fossils, and that's one of the main ideas of paleontology. Paleontology, if you actually Google it, it says study of fossils. So we're going to look at fossils a lot in this class. We're going to look at comparative anatomy and structures. So how something is built and how does it compare to something else that is living or used to be living, and maybe how it's completely different. That's important just as much as how much you're similar. Another one is embryology. So the science of development from one cell into an offspring. And embryology is quite interesting. It seems like it would be an easy topic, but really it's a topic that you can cover for years. And so for us, we're just going to kind of hit the basics, kind of like everything else in this class, because we have somewhere to go, and I want to make sure that we just have a good foundation. And finally, biochemistry. This is a newer one. This is looking at proteins and DNA and comparing them and contrasting them amongst living and non-living, or not, I should say non-living, living and things that have been gone extinct. So these are the four types of evidences of evolution that we're going to look at. These four play a major role in what we're going to be doing in this class. So first thing is fossils. What is a fossil? A fossil is a preserved remain or trace of an organism that is no longer living. So if I go out into the woods and I find a footprint in the ground and it's of a bear, say, I'm in the school forest and it's a bear, that's not a fossil because that animal is still living. Also, it's not preserved. And so we have to make sure we're honest with that. Is fossils have to be preserved, and they're of organisms that are no longer living. They're either traces, that could be imprints, they can be molds, they can be a bunch of different things, but they are no longer living, and it's a preserved remain. So here's some examples here. So we have a couple imprint fossils, a couple teeth, stuff like that. And fossils are usually found in sedimentary rock. And so we talked about the rock cycle earlier this semester. So fossils are found there because that makes the most sense. Igneous comes from volcanoes. You're not going to find a fossil in a volcanic rock. And metamorphic is down below the earth with a bunch of heat and pressure. Fossils wouldn't be able to survive. So most fossils, I would say exclusively fossils, come out of sedimentary rocks. So there's a picture of sedimentary rocks. Now, what's nice about sedimentary rocks is they make layers, and we can use that to age fossils. Like if I can find a fossil right here, well, I know that this much time has passed. So really quick, I just want to make sure that we know the fossil record is not perfect, and we don't have to write these down, but I just want to make sure that we have a clear understanding of them. And we'll talk about the generalities here. And we're going to come way back on fossils and cover fossils a ton later. But there's issues with the fossil record. First, soft tissue is rarely preserved. It's rare for us to find anything that's not bone or imprints. Two, we have covered up a lot of the fossils. It's rare for fossils to form anyways. And that's kind of number three. But a lot of the fossils are destroyed because of what's going on in the crust or what's going on with plate tectonics or movement in local areas. So we don't have as many fossils as we probably would like or should. 3, fossilization doesn't happen everywhere. And so there's spots where, like, there's a reason why no one is digging for T-Rex bones, even though T-Rex lived in Kalkaska area, because the conditions were not right for fossils to be formed here. And so, unfortunately, we don't have any fossils here. And then finally, paleontologists have not dug up every place on Earth. That means we're missing fossils. Like, there's places where fossils probably do exist, but we haven't dug there. And so we're missing some things. So fossil record's not perfect. It's a starting point. Like anything else, you need something as a starting point. Next one, comparative anatomy. So comparative anatomy is the study of anatomical structures to find similarities and differences. What that means is we're going to look at the structure of a living thing or something that has been extinct and compare it to other things' structures. There's 3 kinds of structures we're going to look at exclusively probably in this class. First one is homologous structures. Homologous structures are things with similar basic structures. So they derive from the same ancestor, but they may vary in function. Okay. So for example, one of the most famous homologous structures are the arms in mammals. If you take a look at this mammal list here, actually, sorry, not even mammal list, there's a bird there. Almost looking at almost exclusively all these mammals and then one bird. You'll see that we have different functions for these arms. No one's going to argue that a human arm is going to be used differently than a whale arm. But what you're going to notice is we use the same exact bones. We have a common ancestor with the same exact bone setup, and we just have evolved a different use of these bones. So, for example, we have a humerus. So does the whale. We have a ulna and radius. So does the whale. We have our carpals. So does the whale. We have our metacarpals. So does the whale. And we have our phalanges. So does the whale. And you can see that across all these mammals, same kind of thing. No matter what we're looking at, we are looking at the same structures, just used differently. My favorite's a horse. A horse is actually standing on its finger. It's one fused finger, which is kind of crazy when you look at the actual, like, horse leg, that huge animal is standing on its tippy toes its whole life. That's homologous structures. Analogous structures are structures that have the same function but have different structures, do not have a common descent. So this one's different. Analogous structures is like the word analogy. So I could say, wow, that kid is fast as a river. Well, that's great, but we know that they both run fast, but they're not related in any way possible. They're the same function, they do not have the same structures, and they do not have a common descent. One of the most famous ones is wings. Bugs have wings, reptiles have wings, birds have wings, bats have wings. They have developed in all of those separately from each other. They do not have a common ancestor that has wings, but they've developed these wings and they have the same function. They're all going to be pretty much used to fly or to move through the air and glide, but they have different structures to do that, and they're not a common descent. So same function, wings, but different descent. Another example of this, this is an example of bat and bird. A different example of this would be dolphins and whales and sharks. They both have similar body types. They both have similar smooth skin, fins, whatnot, but they've developed separately and don't have a common descent. And they have different structures to do the same job, even though they have the same function. They both want to swim, but they have different structures to do that. You can see that especially in sharks and whales and dolphins, because whale and dolphins have their tail go up and down while shark goes side to side. The last one, my favorite one to talk about, is vestigial structures. These are reduced body parts that have little to no function, and they're remnants of an ancestor. So vestigial structures is this idea that we still have, not just we as humans, but organisms still have past structures that aren't as useful to them that they share with their common ancestors or ancestors in the past that won't go away because there's really no evolutionary reason to go away. There's so many examples in humans, and we'll kind of read a list this week about them. But one of the most famous examples for us is our appendix. That's a vestigial structure, or goosebumps. They are things that have little to no function in us currently, but they did in the past. There's a reason why they're there. They used to be used, not so much anymore. Another famous one in humans is tail. So here's a picture of appendix. Our appendix is right there. We'll talk about our appendix when we talk about vestigial organs later this week. Oh yeah, we have external ear muscles that we don't use. Some people can still move their ears. Most people can't, though. You might know someone that can move their ear. Our tailbones. There's still people that are born with tails. That's a crazy Google. I don't know if you want to take that Google chance, but it's always interesting. Wisdom teeth is another vestigial organ that we no longer have to use. Penguins, in the examples, their wings. You can see basically going through all of them. This is vestigial structures. Things that have no function in the current living thing, but have a function in past living things. Embryology is the pattern of how things develop, and they can indicate a common ancestor. So, for example, these two are charts. They are accurate charts. This is not a joke. This is not like cartoony. This is the first week or first development stage of an embryo, I should say, because they're all different times. And you can see all these embryos, no matter if you're fish, salamander, tortoise, chicken, pig, cow, rabbit, human, they all look super, super, super similar. And what we see here is we can see a chordate or a backbone forming in all of these right here. You can see the ear hole in all these guys. You can see the eye hole in all of these guys. This is it's called a post-anal tail. The anus is right here on all of them, and the tail goes past that. Obviously, ours shrinks as we grow, and it goes away. And then here are gill slits. These are gill slits. They're supposed to form gills. They don't in most of these things. They actually become other things inside the structure of the animal. For us, our gill slits become our underlying jaw. They also become our vocal cords and set up our cartilage that wraps around some of the main things in our throat. And so our gill slits don't disappear. They just become different things. So, yeah, so they're all pretty much the same all the way through. Okay. And then as you can see, as they go into stage 2, they start changing a little bit, but still these guys right here, you would, if I mix these up, you would have no idea which one was which. And then 3, now you're starting to see a little bit, okay, now we're starting to get somewhere where what they're actually going to look like. But for the beginning here, we can kind of see, okay, look how closely related these are. And you can kind of see, okay, this guy looks more like this guy than he does this guy. And so I'm probably more closely related to this one than this one. And so it's almost like a common sense experiment here. Okay, I'm obviously not that related to this one. I'm pretty far off. But this guy, yeah, we ain't far. This one, we ain't far. This guy, no, it's not bad. Well, these are all mammals. So they should all look kind of similar. So that's what embryology can tell us. And now we can look at biochemical similarities. We can look at the similarities of proteins, RNA, and DNA molecules. We can see how closely related the things are. If they're more closely related, it indicates a common ancestor, indicates that they're more recently separated from each other as a species. Also, this idea that we all have the same proteins, RNA, and DNA molecules. They're obviously slightly different in every living thing, but every living thing can read them. I can actually take code from a bacteria, put it in a human, and the human can read that code. So that shows universality of genetic code also supports evolution within this section. Is if we all weren't having a past common ancestor, we all should be able to code the same information. Also, we all have the same structure of chromosomes. I can't tell the difference between bacterial chromosomes and human chromosomes as long as they're both eukaryotes. We have things that same basic molecules across all of our organelles. This one says chlorophyll, but all of our organelles are all basically the same. So that's a biochemical similarity. So there's 4 that we're doing. This one is kind of hard to see because this is just comparing all the things that make up our cell and DNA and proteins and RNA. We can compare how close they are to each other. Embryology, we're taking a look at how our embryo looks and how they develop compared to other living organisms. Comparative analogy, we're taking a look at how we're structured and how they're similar or different from other living things. And then vestigial is looking at, okay, what do we have now that makes no sense to have, but what was it connected to back in past ancestors? That's vestigial. Analogous is things that have the same function, but they're different structures. So they're not related. So here, bat wings and bird wings, not related, but they have the same function. And then comparative analogy is looking how we use the same structure, but in different ways. And this shows descent. This shows common ancestor. And this would be like the forearm of these guys. And then finally, fossils, they show us remains of past living things, and we kind of use that to build the tree of life. And so all 4 of these come together to give us a look at what we have on Earth, all the different evidence, and how we could have got there. And so fossils get us somewhere. Then the other 3 kind of start filling in the gaps and the holes. So I hope that kind of helps. I hope you did it on this sheet of 4 here. Wrote on every section. I know it was a little long. I'm sorry. But make sure you have all 4. We're going to do this lab. Now, the lab's going to walk you through very similar examples that I gave. Please make sure you read through it. Like I said, it makes you jump from pages to different pages. So please don't get thrown by that. Make sure you're reading it. Make sure you're not just looking at questions. You're reading the instructions as well. So good luck. Thanks, guys, for watching, and I'll talk to you guys soon.